I hope you all would be good. So, here we are back with understanding the last two aspects that we have in case of nutrition in human, and we would be looking into the second last aspect of the nutrition, and that is the absorption with us. Now, you see, the situation that we created in the digestion process was that all of the food particles or the food substances that I eaten up, so when they were there in the lumen of the small intestine, so they were digested into the small particles. The sugars, they got converted into the glucose. The proteins, they got converted into the amino acids, and the fats, they got broken down into the fatty acids, then the glycerol. Now I need to carry them to the cells of my body. But in order to be carrying it to each and every cell that I have got in my body, so I need to firstly absorb these into the blood vessels. If I'm not doing so, then how come the small intestine that we have in the abdominal cavity, it itself cannot move to the brain, to the other parts of the body to be delivering these small soluble food particles that we took out of the particles or out of the food substances that we have eaten it up. So for the absorption, what we know is that this is the process where the digested food particles that we have with us, so they would be absorbed into the blood. And this absorption is taking place there in the small intestine. So the small intestine not only have the role in context of the complete digestion, but also in the absorption of the food particles. Now, here you have to understand is that these are the intestinal walls that I have with me. Here we have got the empty space that is the lumen, and in the lumen, these are the small soluble molecules that I have got. And now, here lies the villi, and through the villi, we would be absorbing these materials into the blood. So, if I were to quickly recall that what we had in the villi verse, so we have got these epithelial cells. Downward the epithelial cells, so we have got these miniature blood vessels, they are the blood capillaries, and then at the center we have got these lacteals with us. And for the epithelial cells that we have here, now you have to be considering as that these cells they have the transport proteins, then these epithelial cells they have the microvilli in them, and then they would have these abundant mitochondria in them. For the second thing that I have got in these villi, so here we have got these blood capillaries. So these blood capillaries, they only have very thin layer of endothelial cells. The word is endothelial. And then at the center, you see, I have got these lacteals, and in the lacteals, we have a fluid, colorless fluid within them, and that is the lymph. And now we would relate that how come the structure of the villi is adapted for the absorption process. Now, if I were to show you the diagram, again, knee diagram. So here we have the small intestine. These are the walls of the intestine. Here we have the lumen. And in the lumen, we have got the digested food particles. I need to take them into these finger-like projections that I have here. And these are the, you could say, the villi with me. And you see from here in the lumen of the small intestine, the particle, they go from the epithelial cells and then ultimately they reach here to the blood capillaries that we have got beneath them. No, we would be like this is one of the frequently asking question that how come the villi are adopted with respect to the absorption phenomenon. So what you have to tell your examiner is that these villi they have got further microvilli on them, and these further microvilli would be increasing their surface area to the volume ratio, so that would make the absorption becoming more efficient. The other scenario is that in these in these villi we have the epithelial cells, and the role of the epithelial cells is that they have the abundant mitochondria, and these mitochondria they would release energy in the form of ATP, and by that the glucose and the amino acid they would be absorbed from the lumen of the small intestine with the help of the active transport. The other scenario is that in the same epithelial cells, we have these transport proteins because the glucose and the amino acid they cannot cross through the cell membranes directly, but they would be requiring the transport protein whenever we have to be moving them from the lumen of the intestine to these epithelial cells and into the blood capillaries. So these transport proteins that we have here, so they would facilitate the movement of the glucose and the amino acids. The other situation was that we have the blood capillaries in the villi, and these blood capillaries in the villi, their walls, they are one cell thick, like you see, they're one cell thick. So this would decrease the distance for diffusion. If we were to say that these blood capillaries that we have here, if they had got multiple cells in their wall, then it would have increased the distance, so the diffusion or the movement of the particles into the blood would become difficult. So that is why we say that the blood capillaries that we have in the villi, so they have the thin wall, and these thin walls they would decrease the distance for the diffusion to occur. And the other situation is that through these villi, the blood it would flow continuously, and when it would flow continuously, so this would maintain the concentration gradient difference between the digested food particles that we have in the lumen and in between the blood that is flowing here. Now, at the same time, you have to tell your examiner that these villi, they also have the lacteals in them, and these lacteals, they have got a colorless fluid in it, which is lymph, and that lymph would be absorbing the fatty acids and the glycerols. Now, the last point is that these, you could say, the kind of the epithelial cells that we have here in the microvilli, into the villi, like these epithelial cells. So you just have to remember one point with respect to the digestion, that the cells in the epithelial, they have got some enzymes in their cell membrane, so that would break down the disaccharides, like the maltose, lactose, and sucrose into the monosaccharides. So in terms of the absorption, you have to remember these points, and then you have to remember these points. But if you have to tell your examiner that what role these villi have in the digestion, so you would say that these epithelial cells in their cell membrane, they have the enzymes that would break down the disaccharides. So here we are done with the absorption thing. I hope you would be clear with this.